Let's dive right into this explainer. Today, we're acting as molecular detectives. We're going to take a graduate-level look at the dense, dynamic machinery operating inside your cells, zooming all the way from the macro-level mysteries of life down to microscopic DNA mutations. We really want to break this down step by step so you can see exactly how your biological blueprint actually works.
So we are starting with a massive question. What genomic information distinguishes a human from a chimpanzee? I mean, we share an enormous amount of evolutionary history, right? Yet here we are, sitting around analyzing our own DNA, and they aren't. Why is that? Well, the answers are actually hidden deep in the sequence of bases that make up our genes and exactly how those genes are regulated.
To get there, here's our roadmap. We'll hit the genome sequencing revolution, the anatomy of the human genome, the power of jumping genes, molecular engines of evolution, what makes us human, and finally, the master builders, Hox genes.
All right, Section 1, the genome sequencing revolution.
Think about the stark contrast here for a second. Sequencing the very first human genome officially started way back in 1990. It was this monumental, highly publicized undertaking that took 13 years and cost roughly $100 million just to read our 3 billion base pair manuscript. But today, thanks to high-throughput, next-generation sequencing techniques, these machines can literally read DNA at a breakneck 25 million base pairs per second. It's wild. What used to take over a decade can now be knocked out for a single individual in a day or so for about $1,000. This revolution has just flooded us with data, basically giving birth to the entire field of bioinformatics just so we can make sense of the whole shebang.
Okay, moving on to Section 2, anatomy of the human genome.
So all that data we gathered, it quickly revealed a pretty fascinating mystery. Humans have this massive genome, 3,000 million base pairs. Yet we somehow survive with fewer than 21,000 genes. To put that into perspective, that is roughly the exact same number of genes as a tiny microscopic nematode worm called C. elegans. How in the world does such vast biological complexity arise from so few genes? Well, the secret lies in molecular tricks like alternative RNA splicing. Essentially, vertebrate genomes get a lot more bang for their buck by splicing the RNA transcripts from a single gene in multiple different ways. This means that one single gene can actually encode for a whole variety of different proteins. Pretty clever, right?
It turns out our genome is way less of a pristine blueprint and much more of a messy, chaotic historical archive. Get this, only a tiny 1.5% of our DNA actually codes for proteins. About 20% are introns, the non-coding sequences inside genes that just get spliced out, but a massive 44% of our genome is made up of repetitive transposable elements. For a long time, this non-coding DNA was totally dismissed as just junk DNA, but we now know it plays absolutely vital roles in genome structure and gene regulation.
Which leads us perfectly into Section 3, the power of jumping genes.
Because of these transposable elements, our genome is definitely not a static textbook. It's constantly editing itself. Transposons basically jump around using a DNA intermediate. But most of our mobile DNA actually consists of retrotransposons. We also have specific families of these sequences, like ALU elements, which make up a whopping 10% of our genome and help regulate expression. Then there are L1 sequences, taking up 17% of our DNA. And what's really fascinating here is that researchers have found L1 sequences to be highly active in the developing brain. They potentially give us the immense diversity of neuronal cell types that we completely rely on for complex thought.
Let's actually look at the molecular mechanics of how retrotransposons move, because they act exactly like retroviruses to copy and paste themselves all over our genetic manuscript. First, a single-stranded RNA intermediate is synthesized from the retrotransposon. Then, an enzyme called reverse transcriptase, which, amazingly, is encoded by the retrotransposon itself, turns that RNA back into a complementary DNA strand. Next, a second complementary DNA strand is synthesized. And finally, this newly minted DNA gets inserted into a completely new site in the genome. Now, this jumping around can absolutely disrupt genes, but sometimes it provides crucial new regulatory functions that give an organism a major survival advantage.
Let's jump to Section 4, molecular engines of evolution.
So, it turns out that genetic errors can actually be highly creative. During meiosis, when homologous chromosomes pair up, sometimes those transposable elements we just talked about cause the chromosomes to misalign. When they cross over, it leads to a genetic cut and paste error known as exon shuffling.
This mechanism literally mixes and matches different protein domains to accidentally create brilliant new proteins with completely novel functions. A really great example of this is the TPA protein, which our bodies use for blood clotting. It's basically a genetic mashup of several different ancestral exons.
We can trace this kind of thing back through time. 500 million years ago, there was just one single globin gene. But because of duplication errors, we ended up with two copies. One became alpha globin and the other became beta globin. Over millions of years, these duplicated again and accumulated mutations. And the cool part is, this didn't destroy the genes. Instead, it created specialized variants, like fetal hemoglobin, which actually has a much higher affinity for oxygen than adult hemoglobin. This specific molecular evolution is exactly what allows mammals to efficiently transfer oxygen to fetuses during pregnancy. It's a real game changer.
Let's zoom back out for Section 5. What makes us human?
When you look at our chromosomes under a microscope, humans have 23 pairs, but chimpanzees have 24. For the longest time, this was a massive puzzle for geneticists, but when we finally sequenced the genomes, we found the smoking gun, a huge structural rearrangement. Human chromosome 2 actually contains remnants of telomeres, the tips of chromosomes, and a deactivated centromere right smack in the middle. It perfectly maps to chimpanzee chromosomes 12 and 13. So sometime in our evolutionary past, two ancestral chromosomes fused end-to-end to create our chromosome 2. Mystery solved.
But honestly, that 1.2% figure is just staggering. If you look at single nucleotide substitutions, just the individual letters of our DNA code, we differ by only 1.2% from the chimpanzee genome. Sure, there are a few more differences if you count insertions and deletions, but overall we are incredibly, unbelievably similar. So how does such a minor difference in the raw code result in a completely different species, one capable of building spaceships and writing complex databases?
That brings us to Section 6, the Master Builders, Hox genes.
To understand this, we have to look at the concept of genetic conservation. Take the homeobox, for example. It's a 180 nucleotide sequence that encodes a 60 amino acid domain inside a protein. This domain binds directly to DNA, acting as a master switch to turn entire batteries of other genes on or off during embryonic development. This exact molecular sequence is so ancient and so insanely valuable that it has been conserved virtually unchanged across radically different animals. In fact, they are so similar that one researcher famously referred to fruit flies as little people with wings.
The similarities across species are just mind-blowing. Homeotic genes, or Hox genes, even exist in the exact same linear sequence on the chromosomes of both fruit flies and mice. This tells us a really profound secret about life. Evolution isn't always about inventing entirely new genes from scratch. Often, it's just changing the regulation of these master builders. Shifting slightly where and when these genes are turned on during development can literally change a crustacean's many legs into an insect's 6 legs. It's those small regulatory tweaks that lead to major macroscopic differences.
And that brings us to FOP2, a transcription factor that regulates vocalization genes. I want to leave you with this final tantalizing thought. Researchers found that mice with their FOP2 gene knocked out completely lose their ability to vocalize. When we sequenced the Neanderthal genome, we discovered they shared our exact human version of this FOP2 gene, which has just a 2 amino acid difference from the chimpanzee version. Could those two tiny microscopic differences in our genetic text be the molecular switch that gave us the anatomical and neurological power of speech? The power to communicate, to build bioinformatics databases, and ultimately, to ask these very questions about our own origins. It just goes to show how microscopic changes create absolutely magnificent complexity.